Philadelphia chromosome. It's a translocation between chromosome 9 and 22. And it leads to leukemia, myelogenous leukemia. So, here is a picture of the karyotype. Here is chromosome 9, here is chromosome 22. And here is a kind of picture artificial cartoon diagram of the picture. So the translocation in the Philadelphia chromosome occurs within a gene. Occurs within two different genes called BCR and ABL. The exact function of BCR we don't know, but ABL is an enzyme. It's a kinase that phosphorylates proteins. And it plays a very important role in controlling the growth of cells. So immediately when you mess around with this protein, growth of cells becomes dysregulated. And what is cancer? It's basically dysregulated growth of cells. And so you end up with dysregulation of blood cell growth and division, leading to cancer. So what happens normally, ABL is a tyrosine kinase. It phosphorylates tyrosine residues in proteins, particular proteins. Okay? And so, in effect, what you end up with is that this enzyme becomes completely unregulated. It starts to phosphorylate everything and allows cells to grow when they shouldn't be growing. So all the breaks and checkpoints that you have that prevent unregulated growth are lost, and the cell begins to grow. Luckily for us, it's a completely new gene. And so pharmaceutical companies were able to design a drug that were able to act only on this new gene, but not on the normal gene. And they were able to inactivate this new gene. It's a drug called Gleevec, G-L-E-V-E-C. And this drug is very specific. It only knocks out the fusion protein, the BCR-ABL fusion that's present in cancer cells, doesn't affect the normal growth of your normal cells. And so it's a very specific drug. And once upon a time, myelogenous leukemia was a death sentence. But after this drug came online, it's basically very, very treatable. So it's not as much of a problem. Okay, but translocations and cancer are very common. There are many, many, many examples where cancers are caused by translocations. And I've just given you a small list of these protein translocations. Of course, here's your ABL and BCR translocation. But you also have other translocations for in lymphomas, in breast cancers, in myeloid leukemias, in prostate cancer, etc. So translocations and cancers are very similar, closely related to one another. And a lot of these translocations are also a function of the three-dimensional organization of chromosomes. So I showed you why inversions take place in a chromosome because of a loop. Correct? This is not in your textbook. I'm just giving this information to you kind of for fun. You don't need to kind of think about it other than from your own perspective and your interests. But translocations, and this is something that's very new. This whole field's developed only in the last 10 years. And that is, your nucleus is not a ball of spaghetti when it comes to the DNA. Okay? Your nucleus is basically has discrete entities. Every chromosome occupies a very specific territory in the nucleus. Each one of you is, in effect, a chromosome in this room, which is the nucleus. Right? Now, you may be close to one another and in certain regions, and then you're far apart in other regions. And the reason you get translocations is because you have two breaks. A break of an arm in one person and a break of an arm in another person, then you mistakenly swap segments. But it only happens because you're sitting close to one another. If you were far apart and you had a break of the arms, you wouldn't be translocating unless the arm flew from one end to the room to the other. And that's why translocations happen very often between very specific chromosomes. The reason is those chromosomes reside close to one another in the nucleus. And that's why you almost always get certain translocations more frequently than any other translocation. This is a picture of a nucleus, human nucleus. This is the cartoon diagram of all the data that was generated. And you can see each one of the chromosomes occupies a very specific territory. And I can't find 22 and 9, but I'm sure it's there somewhere, probably behind the picture, and they reside close to one another. And because of that, you very often get this specific translocation. And this is just for your interest that I'm showing you this picture from a paper. All right, so we're done with gross chromosomal rearrangements. Any questions? There are 4 of these that you have to think of: deletions, duplications, inversions, and translocations. All right? And the consequences of those mutations when cells go through meiosis and generate gametes that will get fertilized and produce progeny. That's what you guys have to think about. All right? Any questions on this? This is all chapter 13 in the 4th edition. Okay. We've got 4 more lectures to go, 12, 13, 14, and 15. In, I don't know how many actual lectures do we have left? How many? 5, 6? Does anybody know how many more weeks left of this quarter? 6 more. So we've got 6 lectures, we've got 4 lectures to do in 6 times 2, 12 hours basically. All right. We almost never get to the last lecture on the lack of run. So it's entirely possible we will this year. But you guys aren't asking a lot of questions. Don't just start asking questions and show me now. But I love the lack of run. It's a paradigm for gene regulation. Okay, so we're studying mutations, right? And we've been focusing on gross chromosomal changes. And we've been saying, how do we study them? Well, we study them under the microscope. Deletions, inversions, insertions, or duplications, and translocations. And we basically are studying them using the microscope. But now what happens if you have an extremely small mutation? Tiny deletions, tiny additions. How do we study those? That's where we now move. So before we follow up on the method to study this, I want to first talk about the small mutations and the consequences of those small mutations. Once we've talked about those small mutations and their consequences, then we'll talk about the methods involved in the study of these small mutations. So we are back in chapter 7 for a little while, and chapter 8. Chapter 8 is simply background material that you absolutely need to know in order to answer these questions, which is transcription and translation. Okay. So we're going to talk about small deletions, small insertions, and point mutations, i.e. a single nucleotide change in a gene or in anywhere in the chromosome. Okay. And the methods we use are all going to be kind of grouped together in one big title: recombinant DNA technology or gene cloning. Now, one thing to keep in mind, gene cloning is not the same as animal cloning. You probably heard of Dolly the sheep, correct? They cloned the entire sheep. That's totally different. Okay? That's somatic nuclear transplants. We're not going to talk about this. We're talking about gene cloning. Define cloning. How do you guys visualize? What's the meaning of the word cloning? An exact copy of something, correct? Many, many copies, or 1 copy, or 2 copies, or whatever. So we are going to basically clone an exact copy of a piece of DNA. That's what we're talking about when I'm going to talk about cloning and recombinant DNA technology. We're not talking about the cloning of an entire organism, just a single gene or a single segment of a DNA. It's a fragment of DNA. Okay, so most mutations are harmful. And we've seen this slide. I've gone through this already once. I'm not going to go through it the second time. I don't know why I have it there. Let's first look at tiny, tiny, tiny mutations. Okay? Missense mutations. What's a missense mutation? This is the first class of tiny mutations we'll focus on. I've just swapped this around in my notes. I think 2 pages or 3 pages later you have missense mutations. I just moved them up 2 pages earlier. That's all I've done. So flop back and forth in your notes and you'll find this. So a missense mutation alters 1 codon. And it's not even 1 codon, it's 1 nucleotide of a codon. Very often. Okay? And the result of this is it encodes a different amino acid. Right? Okay. So let's look at it. Here is our sequence of the wild type gene. Something simple. U, 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 G, C, U, 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 U. That's the messenger RNA sequence of the wild type gene. If you have a missense and you see a code for a protein that will go phenylalanine, phenylalanine, cysteine, phenylalanine, phenylalanine, based on the codon sequences. Right? UUU codes for phenylalanine, UGC codes for cysteine. Okay. Now you have a mutant, and the messenger RNA codes UUU, UUU, but it goes UGG, UUU, UUU. This C over here has converted to a G. That's an example of a missense mutation. Because when you translate that RNA into a protein, you get a protein that will be phenylalanine, phenylalanine, tryptophan, phenylalanine, phenylalanine. Because UGG codes for tryptophan, UGC codes for cysteine, and so your protein will have a mutation in it. So you'll have converted your protein, there will be one amino acid that has changed. Only one amino acid has changed, not all of the others. The other amino acids remain the same, it's just this one amino acid that has changed. And this is an example of a missense mutation. You're changing the sense of that one codon. Right? That's what this is. Okay, so what are the consequences of missense mutation? What happens when you have a missense mutation? They're hard to predict, is a simple answer. You have to do additional experiments to figure this out. I'm going to give you a few examples of what might happen when you have a missense mutation. So, missense mutations alter one out of the many amino acids of a protein. And so, the consequences depend on which amino acid is altered. Let's imagine you have a mutation that converts a lysine to an arginine. How many of you have taken 28? Everybody, right? Good. What's the charge on a lysine? Plus or minus or none? Plus. Lysines are plus. What about arginines? Plus, minus, or polar, non-polar? Plus? Yes. Both lysines and arginines are positively charged amino acids. So when you convert a lysine to an arginine, you're not really changing the charge. You're changing the side group a little bit, not dramatically. And because of that, the protein can tolerate these mutations quite well. If you make a change from a lysine to an arginine, most of the time, the protein functions normally. Occasionally, you have a mutant phenotype, but most of the time, it doesn't. Similarly, with a change of glutamic acid to aspartic acid, it's usually conserved because you're changing a negative to a negative, and it's fine. Similarly, with leucine and isoleucine, or leucine and alanine, these are small polar changes. The cells can adapt to it. Proteins are not misfolded dramatically, and they function quite normally. So what happens when you have a change that looks like this, where you go from a lysine to a glutamic acid? Now you're going from a change that's positive to a change that's negative. And under those conditions, proteins tend to completely lose their function. So just a one single amino acid change, very often, not always, but very often, can lead to dramatic changes in the function of a protein. And so you obviously can't tell this just by looking at the change. You can predict to some extent. Probability is higher that the protein will be mutated when you have this change than this change. But it's not 100%. You have to do the actual experiment to figure that out. But higher probability. The other important thing is, where does the change take place in the protein? Proteins are three-dimensional molecules, correct? They're enzymes or structural proteins, but they are three-dimensional. They have a certain shape. There are amino acids in the interior that hold the protein shape together. If you have mutations within the interior, especially mutations that convert lysine to glutamic acid, that internal shape is going to completely disrupt. The whole protein is going to denature, and it will lose its activity. If you have a change on a tail that's kind of unstructured, maybe a lysine to glutamic acid may not have a big effect. So we don't know what's going to happen until we do the experiment. But the location of the change has a consequence on whether you have a mutant function or a wild-type function. Mutations in globular domains versus unstructured things. That's the other point. Whether you have a mutation within a structured region or an unstructured region. Structured regions, all the interactions are important to maintain the structure. If you have a mutation that alters those interactions, protein structure might change and then its function change. So, another aspect to think about. And then there are certain classes of mutations that are silent, i.e., there is an actual mutation in the DNA, but that does not result in a change in amino acid sequence because of the degeneracy of the genetic code. And because of that, these mutations are called silent mutations. There has been a mutation taken place. It just hasn't changed the amino acid sequence, and so you don't really care. And then mutations in regions that are not coding for proteins. Now, these may be silent mutations, or they may not be silent mutations. It depends on where the mutation is. So if this mutation lies in a region that is important for enhancer function, it might affect the expression of the protein. If the mutation is in the promoter of a gene, it might affect the expression of the gene. And therefore, it may also have a phenotype. So mutations in non-coding regions can or may not have phenotypes. It depends on where they lie. Most of the times, they have no phenotypes because the vast segments of your genome have no function. They're just sitting there as a buffer. And so it's useful to have mutations in those regions. Okay. And then there's one class of mutations we'll come to later on called nonsense mutations. These are tiny mutations, missense mutations, that convert a codon to a stop codon. So you're going from a codon to a stop codon. So you're changing, you're putting in an artificially putting in a translation stop site. That's known as a nonsense mutation. Okay. Another class of mutations we're going to talk about are called frame shift mutations. Frame shift mutations are mutations that either insert 1, 2, or 3 nucleotides, or a few more, or delete a tiny number of nucleotides. So they basically, frame shifts are insertions and deletions of small numbers of nucleotides. Okay, so a single base pair deletion or a single base pair insertion will result in the change in the reading frame of a protein. How does that work? Well, let's imagine here is your reading frame. Okay? Some sequence NNNNN, and then you have AUG, UUU, ATC, UUU, etc., etc. So this is your RNA being made. A sequence that runs all the way through. Okay. So let's see what happens. So this will code for a protein that goes methionine, phenylalanine, serine, phenylalanine, serine, phenylalanine, serine. Now, you're going to delete a single C. You're going to delete this cytosine in this protein. In this messenger RNA. Here's what the sequence looks like. The sequence hasn't changed dramatically. It's the same. Here's your AUG, the UUU, and then you have the A and the G, and then the C is removed, and you bring in the U, UUA, GCU, UUA, GC, and so on and so on. But you remove that one nucleotide. Right? So now, let's imagine the protein is being made. What does AUG code for? It codes for a methionine. UUU, phenylalanine. AGU is a serine. So AGC is a serine, AGU is a serine, so it hasn't changed. But notice what happens after that. UUU coded for phenylalanine. Now, because of this removal of a single C, the whole frame of the sequence has shifted. And now, the sequence downstream of this mutation is very, very different. Right? So that's a major consequence. So a frame shift mutation has major changes in protein sequence. Downstream of the mutant site. Not upstream. Upstream of the mutant site, there is no change. It's only the minute you go past that mutation site. Because you've removed one nucleotide and the genetic code has no commas and no overlaps, you shift the frame of reading when you're reading through the ribosome, and you end up with reading proteins. This is with respect to a deletion. Let's imagine that in this sequence, instead of deleting the C, you're going to insert a C into the wild type gene. That also has major consequences. So right after this C, you're going to insert a second C. So here's the sequence, AGC. That's the same as the wild type. But now you've inserted a C, and so you go C, UU, UAG, CUU, UAG, C, and so on and so on. And when you translate this protein, you get methionine, phenylalanine, serine, and that's fine up to here. But then you have the additional C, and that makes it a CUU codon, and that CUU codon, instead of phenylalanine, you have a leucine. But now because of the frame shift, you get a UAG, which is a stop codon. And so you truncate the protein. So an addition of one nucleotide, a deletion of one nucleotide has major consequences on the protein sequence. Okay, so single base pair deletion or a single base pair insertion results in the change of the reading frames. So remember, a single molecule of DNA or RNA has 3 different reading frames, correct? One molecule has 3 reading frames, the other one on the other side has 3 reading frames. So here is your first reading frame: methionine, phenylalanine, serine, phenylalanine, serine, phenylalanine, serine. What has happened is there is a second reading frame that would go cysteine, leucine, alanine, leucine, alanine, leucine, alanine, etc. And when you deleted one nucleotide, you shifted from this reading frame to this reading frame, and so you changed the sequence. So that's what we call a frame shift. You've shifted the frame of reading, basically. And when you inserted a nucleotide, you basically went to something that looks like this: leucine stop, leucine stop, leucine stop. And so that's what's going on. You're shifting the reading frame once you start with a single reference point, which is the A and G codon. So when you delete it, you go from one to another. You go from methionine, phenylalanine, serine to leucine, alanine, leucine. So you go from methionine, phenylalanine, serine. You switch to leucine, alanine, leucine. You shifted a frame. When you insert an extra C, you go from methionine, phenylalanine, serine from this frame to leucine and stop, which is this third frame. So that's why these things are called frame shift mutations, because you're shifting the reading frame. Now, let's talk about, you delete a single C. Here is the wild type sequence. You get phenylalanine, serine, phenylalanine, serine. Now, you delete this C. Okay. Let's delete the C and see what happens. That's the same as before. Methionine, phenylalanine, serine, leucine, alanine. What happens if you delete 2 nucleotides, the G and the C? When you delete 2 nucleotides, the new RNA sequence looks like this. So you deleted the G and the C, this A is still there, but then these U's shift up, and you become AUU, UAG, CUU, UAG. When you translate this protein, you get a sequence that goes methionine, phenylalanine, isoleucine, and a stop. So one nucleotide change is a frame shift. 2 nucleotide change is also a frame shift. So there's a problem. What happens when you delete 3 nucleotides? When you delete 3 nucleotides, A, G, and C, you end up with AUG, UUU, UUU, AGC, UUU, AGC. And when you translate this, the sequence is methionine, phenylalanine, phenylalanine, serine, phenylalanine, serine. What has changed? You've lost 1 amino acid in this protein. That's all you've changed. So a loss of 3 nucleotides, the cell can manage often. A loss of 1 or 2 is really bad for the cell. So multiples of 3 are fine. because you're only removing 1, 2, or 3 amino acids. But when you have insertions of 1 or 2 nucleotides, or multiples of 1 or 2, then you have problems. Yes? They call small deletions at that point. They're not a frame shift. Questions? No? Okay. So we carry on. So let's look what happens when you insert multiple nucleotides as well. So here's the wild type sequence. Looks for codes for the protein, methionine, phenylalanine, serine, etc. You insert a single nucleotide, you basically change the frame, and you end up with a truncated protein with a premature stop codon. You insert 2 Cs, you now inserted 2 Cs here right after this HEC, and you have a similar problem. You have a totally different protein sequence downstream of the site of insertion. So instead of phenylalanine, serine, you have proline, leucine, alanine, leucine, etc. But then when you insert 3 nucleotides, you basically don't change very dramatically. By putting 3 Cs in, you've inserted an extra amino acid, a proline, but that's the only thing you've done. You've added 1 amino acid. The rest of the sequence of the protein is more or less unchanged. So once again, addition of 3 nucleotides also is not dramatically problematic for a cell, like a deletion of 3 nucleotides.
Question?
Yes?
What if they were added at different positions?
Very good question. What happens when you have them at different positions? So you're basically asking, if I insert 1 nucleotide here, 1 nucleotide here, and 1 nucleotide here, then the amino acids in this segment will change, but everything on either side of it will be normal. And it has to be a multiple of 3 for it to be normal. If it's not a multiple of 3, you can have a 1 nucleotide addition here and 1 nucleotide addition here. They'll still be frame shifted all the way through basically. Same thing. Same thing. So the sequence within this region will change, but nothing outside. Now, when the sequence within it changes, if by chance you have a stop codon, then you're dead in your tracks and you're going to have a mutant protein. So you have to basically solve the sequence and figure out what the consequences of that change, those different changes are going to be on that protein. Okay, so what are the consequences of silent mutation? I'm going to show you an example of a silent mutation. Here is the wild type sequence. But the point to remember is the genetic code is degenerate. There is more than 1 codon for the same amino acid. And so here's a wild type sequence. You can have a point mutation that converts UUU to UUC. That's the point mutation. It's a change of a U to a C. When you translate this, you find that there is no change in the protein sequence, because UUC also codes for phenylalanine, just like UUU. And so this is an example of a silent mutation. There's no consequence, so it's silent. Okay? Mutations that occur within introns are also silent, because introns are spliced out, so they have no consequences. Right? Mutations that occur in regions that are not part of a gene are also silent, because they are in regions that have no function. And so those are also silent mutations. Now, just because they're in a region that is not coding for a protein doesn't mean they're always silent. Because there are regions of the genome that don't code for a protein, but do have functions, like the promoter, like the enhancer, like a splicing site, etc. And so those will have consequences. So mutations in non-protein coding regions of a gene will have consequences. So mutations in the promoter of a gene, in the junction where splicing takes place, or in a ribosome binding site, or in an enhancer of a gene, all of these will be mutagenic. But you can't predict the way it will work. You may have increased production of a protein, you may have decreased production of a protein when you have these mutations. And so you can't determine what's going to happen. You can have reduced expression of messenger RNA, which may result in reduced levels of protein, and that may have no consequence. The cell may be happy with 5 molecules of protein instead of 10 molecules of protein, because usually enzymes are in excess, so it may be not problematic. Alternatively, it might have a problem. So you have to figure this out. You can also get mutations in genes that make